Oh, yeah. Hi. Okay, this is another in our series of ISET tutorials, and I'm David Brainerd, and with me here today are, I'll let them introduce themselves.
Brian Wandell, here in California.
I'm Joyce Farrell here in California as well.
I'm Nicholas Coleris with David Brainerd at Penn.
Great. Okay, so this tutorial video, I would say, is one that you might watch after you've watched some of the earlier ones we recorded, including the ISETBio overview, which we called RGB imaged excitations, which gave an overview of calculating things in ISETBio, and then Brian recorded tutorials on ISET scenes and ISET optical images, which are core structures and computations that we use as we move from the external stimulus back toward the imaging plane. And what I'm going to do today is really talk about how you set up optical images and a cone mosaic in the imaging plane to compute excitations, cone excitations with realistic models of the human optics and how some of the properties of that can be manipulated. So, and I'm going to do that in the context of this script, which is available in ISETBio called T_C mosaic and optics off axis. And we'll kind of go through that, and I'll go back and change some pieces. So just to get us going here, we would typically start by running IE init and clear and closing any extant figures. And the first thing I'm going to do, which is not something I'm going to spend a lot of time on here, is I'm going to create a checkerboard scene using the scene create command. And you can see the scene tutorial for more about scenes and how to create them. I particularly set it up here so I define an overall field of view for the scene, and I define a size in degrees for each check in the checkerboard and a number of pixels. And then from that, I compute how big the check needs to be in pixels in order to pass that to the scene create. And also it specifies how many pairs of checkers, white and black, do you get, and that is calculated to come out to the overall size. So I'm going to execute that, and we'll take a look at that scene, but not dwell on it, just so we sort of see what we're working with here. Okay, so here's a, go ahead, Brian. Yeah.
I was just going to say, it's very cool that here you've created a whole spectral scene. It looks black and white, yeah, but there's a whole spectrum there, and all the units are known.
Yes, and indeed, in this scene window, we can see it's from 400 to 700 nanometers at 10 nanometer increments, and I believe this is a equal number of photons per second in each wavelength band.
If you click on that bad boy, it'll be
right there.
Come up for us. Yeah, just click on it. It'll come up as a window, and we can see it.
Oh, look at that! All right. So equal photons and some other information about the scenes. This one has a mean luminance of 100 candelas per meter squared, and other little facts here for you. Okay, so we've got a scene, and now I'm going to specify some things I want to control in this tutorial that we'll be using. I want to specify where on the retina, with the fovea being at 0, 0, and the first coordinate being the horizontal position, where do I want the mosaic that I'm going to, the bit of cone mosaic I'm going to create to be centered in degrees. And then I'm going to say some things about the, we're going to build an optical image structure to model the human eye. And so there's some things we need to say to do that. I'm going to accept a number of default parameters. But you may be familiar with, the human eye has a chromatic aberration, lateral chromatic aberration particularly. And normally when we build a human eye optical image, we have that turned on. And but I will later show you what happens if we turn it off. So there's a flag that's going to control that. The human eye is aberrated. It has optical aberrations, and we're going to talk in a minute about where we get measurements of those. But we could also consider if everything else was the same as a human eye, was it diffraction limited? So I'll come back to that. But diffraction limited human eye is false. We'll start out working with a 3 millimeter pupil, which is sort of a nice daylight size pupil. And we'll assume that the eye is in good focus on whatever it's looking at. But for various modeling purposes, we might want to ask what would happen if we added some amount of defocus. And so that's one more thing that the script below is going to control. So those are some parameters we'll look at in terms of the optics. Plus, we've got to get measurements of the wavefront aberrations that describe the human eye. And Brian talked a bit about wavefront aberrations in the optical image tutorial. For the human eye, there are very nice sets of measurements that have been made. One set made available from Larry Tybos's lab as early as 2002, with quite a few eyes, but only in the fovea. And then in 2012, Pablo Artal published some measurements across the horizontal meridian in, again, quite a few subjects. And then Pullins and Artal, again from Pablo Artal's lab, published measurements on a grid across the retina. So these data sets allow us to explore how the optics change as a function of visual field position, which we will do a little later. But we have code and wrapper code set up, that I'm going to use here, that allows us to select any of these three databases, which we've curated as part of ISETBio, specify one of them, and then they have various subjects in them, and we've done some work to evaluate the quality of the optics. And so from best to worst, you can select a subject from each of these data sets. And if you put in a number larger than the number that's actually available, which varies across there, you'll get the worst subject in that database by a summary measure of optical quality.
So David, let me slow you down just a little bit. I find this part just really inspiring in a way. I've just been back and forth with Artal, telling him that it was great how he contributed these measurements. I just think it's worth maybe noting that basically these guys are giving us the point spread function of the optics for different parts of the visual field. So when you say on 0, 0, that means you're asking for the point spread function measured by one of these groups right over the fovea.
Yep,
And yeah, and the Larry's data, the Tybos data, are all from on the fovea, and it's quite a cool thing that Artal and his group, the Pullins paper is very nice. It just actually measured it at different positions, because the human eye as a whole is not shift invariant, but over local positions it is.
Another thing I should mention, Nicholas did spend some time with these various data sets, which, as you say, it's great that have been made available. And it's very pleasing that if you go to look at the quality of the foveal point spread function across all three data sets, it's quite consistent. So I would say that our conclusion was that people in physiological optics, at least across labs, can make measurements that are not massively contradictory and sort of build together to give us the bigger picture. Yeah. And just to comment on your comment, as part of this ISETBio project, one of the things that really has been critical to do most anything are the data that have been collected mostly in other labs, not in our lab, that we've been able to use to calibrate the software. And without that, it just wouldn't be possible to sort of have this image computable model. So we're very appreciative of that.
Yes, and let me add, and I'm also appreciative of Nicholas, who did go through the data, and they were great. But he found some stuff, and it's been curated, and somebody might go after our data and curate it a little further, and let us know if you do.
Yeah, yep. The gist is rather subtle here, but these are our curation of the data sets. Okay. So we're going to specify, and also some of these data sets are right eye and a left eye data. We're going to specify which eye we take it from. And let's see, I wrote some of these. So we're going to use the Pullins 2015 data for most of what I do here. And okay, so I'm going to run down from wherever I stopped last, which might have been about there, and execute down through here. I'll unpack a little bit, just some bookkeeping that we're really doing in this switch statement. Each of these data sets is implemented as essentially a class, and the ranking that Nicholas worked out across the subjects can be returned, and then we check to make sure you didn't ask for a subject who was longer than the data set, and if you did, we give you the last one in the rank order. I think I've done that right, and otherwise we actually, I should probably have done this wrong, right? It should be ranked subject IDs This is a bug. I haven't tested this. I'm going to fix it. It should be ranked. IDs, the last one, and I'm going to fix that for all 3 data sets. So it's right if you go so far as to run this tutorial. I didn't test that particular bit of code. And then some of these subjects, we found that there was quite a bit of residual defocus in the measurement table. And when that was large, we set a flag or have a flag that tells us, maybe you'd like to stimulate the optics of this subject, assuming that they weren't, that they were properly accommodated as opposed to the state they were when the optics were measured. That wasn't true for all the subjects. And for those, we assumed the residual defocus was sort of their typical viewing. So that's just a little bookkeeping.
Question.
Yeah.
For how many subjects are there, and does the ranking have some kind of meaning?
There are varying number of subjects for the database. The Poland, I think we've got 10. The Artau 41, and Tybos had 200, but we seem to have curated about 70, at least for the right eye. And Nicholas, can you remind us what figure of merit you used when you ranked subjects?
Yeah, it's how concentrated the PSF is. So I think it's the product of the sigma along the x and along the y-axis.
Basically, how sharp the image is.
How sharp the image is, but how concentrated the PSF is at the in focus wavelength. And I should say these are measurements of the monochromatic aberrations. We're using standard tables to build in the chromatic aberration that.
So there's going to be some student out there who's going to be wondering whether this is closely connected to the famous Strel ratio. And the answer is yes, it's very closely connected.
Yeah, it is. Yep.
Yeah. There are lots of okay, maybe we won't belabor that longer. Okay, so now as we talked about in that RGB image excitations tutorial, we're going to create a cone mosaic object, and we're going to accept its default parameters, except for its size, which we're going to specify to match the size that we built the scene at. And so we give the size in degrees, and then we're going to say where on the retina we want to pull it from. So we're going to take the parameter we set up here. And so that's going to be our mosaic. I'm going to create the cone mosaic. That could take a second because it loads pre-computed data. And there we go, okay, but not too long. And now the particular way, there are lots of ways to build these optical images. I'm going to show you a wrapper method that is actually part of the cone mosaic class that knows how to build optical images that are matched to the eccentricity that we built our cone mosaic at. And so we're gonna build an optical image ensemble, and I'll tell you why it's an ensemble in a second, but we're gonna only use one of them. And we're gonna tell it that we want to use the database that we specified up above, and the subject ID and the pupil diameter. Spatial point spread function sometimes is measured or shifted quite a bit, and we find it a little bit more natural to just think about them as having their center of mass along the optical axis. So when this is true, we shift them together across wavelengths back. This is that fixing up of subjects who we thought might have been a little out of focus. And then there's a calculation that goes from the raw optical measurements, which are measurements of the so-called pupil function or the wavefront aberrations, which get specified on a grid. And I'm doing that on a 201 by 201 pixel grid here. So I'm going to let that rip, and then I'll just say that this same method, if we wanted to compute what happened for an eye movement path, where we imagine that the eyes were moving, you can pass to this same method an eye movement path, which I'm not going to illustrate today, and it would generate one optical image shifted around to model the eye movements, and therefore the shift of the optical image on the retina, but not doing that here. And so all this comes back as a cell array of optical image objects. When I haven't put the eye movements in, there's just one of them, and I'm going to dereference it from the cell array and just have an optical image. which is again the structure that, or the object we talked about, Brian talked about in the optical image tutorial. So now we've got, did I run this? If not, I'm just going to run it again. Be sure. And okay, so now we've got an optical image, and I just want to check something that's easy enough to mess up, and there's lots of things you have to worry about. Is you could have specified the focal length of the eye in the cone mosaic, and you could have specified it in the optical image, and you sort of want them to be the same. And I believe we've set this thing up, or in any case have the default right that they're the same, but I actually got from the optical image the focal length, and I converted that to a size, an estimate of the microns per degree, and I made sure that as a fractional error, the cone mosaic and the optical image agree about how big the eye is, since that's a parameter that each of them could be varied, and we're good. So that was just a little check.
And I would say David and Nicholas, and me too, but these guys all the time are doing quantitative checks in and out, and that's really important. And another way you can check it if you're a beginner and you're not quite sure what to multiply and so forth. Some of this stuff, if you just bring up the OI in the window, you'll see the numbers, and that might be, might be worth remembering. So
I'm going to bring up the OI eventually, but let me just illustrate that you really can do that, except I haven't computed on it yet. So no, I could do both of these things. This is I would call this the undoctored OI. I'm going to do some potentially do some manipulations, but let's just look at this. Here's the checkerboard, and it's got moderately coarse checks. This is, I think Brian just taught me that I can double click this, and there it's going to pop up, and we can see things like that it's a shift invariant optics, so it's a point spread function, and it's wavefront optics were built on it, and got this focal length, and its F number is this, which comes from these two parameters together. There's our 3mm pupil. It's right for the human eye. Particular choice of how we're computing things. It's rows and columns, and it's horizontal and diagonal field of view, which matches the scene. The wavelength sampling matches up. That's good, and it's got height and width, etc. So that's what this looks like, and we'll come back to this a little more in a minute. In fact, I'm gonna.
Yeah, there's some student out there wondering why that's yellow and purple, and you're going to explain
and
that.
Why is it yellow? Well, somebody out there can tell me. I was going to explain it at one point, but we could say it now. I guess I'll say it now. So one of the things that we have modeled in this is that it was not true in the other video, is that the lens of the eye is absorbed selectively and primarily in the short wavelengths, so that as light passes through the lens to the retina, it becomes colloquially yellower or has less short wavelength power. And we model that when we compute the retinal image for the human eye. We have the lens absorption baked in there. That's a parameter you can turn off if you want. I'm not going to illustrate that today. And if you're familiar with the macular pigment, again, I won't say much about it, but the cone mosaic object also has the macular pigment, which also absorbs in the short wavelengths over the central bit, modeling Maxwell's spot in the presence of this inert pigment.
And one of the things we're happy about, is, people kind of hear this stuff in the air. Did you worry about the macular pigment? Did you worry about the lens? Well, we worried about that for you. So, there you go.
Yeah. Great.
Nice.
All right. So I'm going to close this now. I sort of want to say a little more about how we got here, or at least some of the things that we could do on the way, because after I go through these things that we could do on the way, I'm going to show you some results for this setup. And then what we'll do for the second part of the tutorial is really just explore what happens to various pieces of the calculation if we change stuff around. So we did this check, and now I'm going to actually get out of the optical image. I'm going to get the wavefront object, which has the pupil function and the wavefront aberrations, and I'm going to get from that the Zernike coefficients, which are the expansion that describes the wavefront aberrations. And again, I think Brian talked about these a bit last time. For the human eye in this data set, let's see how many we've got. There are 30 of them, but a bunch are 0. And these are just describing, each of these is the weight on a little function over the pupil plane, which describes how white light is accelerated or retarded each position in the pupil, which is a way of describing the aberrations. And these came from that Poland's data set for this subject, for this retinal location. And I'm not going to, again, do more about unpacking that in this tutorial, other than to say they're there, and also show you what we would do if we wanted diffraction limited or to change the defocus. Then the wavefront object itself specifies whether you want to do something about lateral chromatic aberration in this field called the LCA method. And you can see here that the LCA method is human, which means it's going to use standard data to control the lateral chromatic aberration of the eye. And if we set that turn it off flag there, we would just set that LCA method to none, which is what you get when you build a optical image without specifying that it's for a human. For example, if you're modeling a camera where the chromatic aberration would be far less.
So just a note about that. For me, over many years, I thought there was complete consensus on what the human lateral chromatic aberration is. And I put that in my book as, oh, here's what, everybody kind of agrees on this. And in recent years, partly from Austin Rorda, Alf Dubra, David seems to agree with these folks. There seems to be a little more variation between people. The mean people agree on, but the idea that there's a little bit more variation between people is something that's a recent development, and we're thinking about what to do about that in this code.
Yeah, and we actually have worked with our colleague Derek Nankivil, Nicholas, and I, and published a paper that built on Austin Rurda and Derek and Marty Banks's work that involved curating data and measuring chromatic aberration across individuals. And we have online and in a usable way point spread functions that actually model some individual variation in chromatic aberration, as well as transverse chromatic aberration, which we're not modeling in this code here. So that's something that's possible to do, described in a recent paper in, I think, Optics Express that Derek is the first author on.
So I have just a quick question. You probably don't want to do this now, but maybe you could point people to a reference. The difference between transverse and lateral chromatic aberration. Maybe there's a reference you can point them to. And what's nice about this is you could see that you could determine whether or not it makes a difference, or how big of a difference it makes.
Yeah. So lateral chromatic aberration, or sorry, did I say lateral? I really meant longitudinal. I should have been saying longitudinal here. Right. I should have been saying longitudinal. If I've been saying lateral, it's just because the L took over my brain. But longitudinal chromatic aberration, which is also called axial chromatic aberration, is a defocus that comes from the fact that the refractive index of the lens and cornea depends on wavelength. And transverse or lateral chromatic aberration, transverse is probably a better word, is a shift, a wavelength-dependent shift in magnification of the image at each wavelength that depends on that refractive index change, but interacts in detail with the shape of the lens and exactly where the pupil is and whether it's on center axially and laterally. And we have to go to an optical engineering text to see that in detail, although you can ray trace it, and some of the work Brian referenced with ISET3D might allow that. But that's what the two things are, and here we're just focused on the longitudinal. Okay. Did you want to say something, Brian?
No, I put it on mute, but these are good topics for a, we'll do little tutorials on this stuff.
Great. The other thing that one might want to do is ask, well, suppose the human eye weren't aberrated and were diffraction limited, and certainly our colleagues who work with adaptive optics and do psychophysics with adaptive optics sometimes correct for longitudinal chromatic aberration in their stimuli, which is one reason you might want to turn this off. And they also correct for optical aberrations in the stimuli that they're presenting to human subjects. And you can model a otherwise human eye with diffraction limited conditions by setting all the Zernike coefficients to 0. So that would be done here if this flag were true. And then finally, even in an adaptive optics system or in various other situations, you might want to wonder what happened if there was some defocus. And so we had added defocus diopters, which is currently set to 0, to add it directly into the Zernike coefficients, which I'm showing here. Shame on me for hard coding it as the 5th entry, but I happen to know that's what it is. And there we add it in microns because that's the units the Zernike coefficients are. So we convert the defocus and diopters to microns, which depends on the pupil diameter. And then having done any of this stuff, we set those Zernike coefficients back into the wavefront structure. And I get to here, I'll plot some point spread functions at different wavelengths just to show you how different they are under various different conditions, particularly the chromatic aberration. So these will be plots we'll look at. Then we're going to put the wavefront structure back into the optical image, and we're going to compute what the retinal image looks like and look at it. So I'm not going to do that whole set of stuff, which I sort of skipped over from here. And I think we're going to get the same image we have because I have everything turned off. But we should have, oh, okay, great. So here we have a couple things that are worth enjoying. One is so that the eye's optics, the human eye's optics are aberrated. This is a 3 millimeter pupil, and you can see that although here's the point spread function at the in-focus wavelength, this position on the retina in microns in 2 dimensions, and this is now the basically the height of the point spread function under circumstances where volume integrates to 1, which means all the light has to go somewhere. And as you might remember, 300 microns per degree, more or less, 280. This is 80 microns across. So we could call this a 1/4 of a degree, give or take. And so the point spread function is quite compact at the in-focus wavelength with a 3 millimeter pupil, but it's a little bit aberrated there. And then you see the enormous effect of chromatic aberration, which is basically defocus interacting with the aberrations. Here I'm at 400, and this point spread function is spread out to occupy almost a 1/4 of a degree. And here I'm at 700, and it's in between the two. The change in focus from chromatic aberration is more severe per nanometer going to the short than it is going to the long. But that's going to cause a wavelength-dependent blur. And I mention that because we should be able to see it here if we went to this window and use the technique that Brian described last time we could do to make a little plot of slice through this this thing. What have I done? Here we are with position. So it's the checks on and off and on and off. And then you see, oh yeah, I've done exactly what I thought I've done. You see here we've got wavelength, and for each wavelength, we have this slice in the retinal image. And what you see as the modulation goes at 400 nanometers, where there's this enormous point spread function, there's almost no contrast. There's also less light because this is an equal, we're plotting the irradiance in, actually, I don't quite understand why the irradiance is going down, because I thought it was an equal quanta spectrum, but maybe Brian knows. Wasn't our scene an equal quantum spectrum?
I think
Anyway,
it's equal photons. Yeah, the initial input was equal photons at every case,
yeah.
and then the lens would have blocked a lot.
Oh,
of
it's the lens. It's the lens. Thank you. I knew I figured this out earlier today.
But we're always doing
lens.
this to one another, by
Yeah.
the way. This
Yeah.
is kind of normal stuff. Why is this way? And we're checking
Right.
and double checking. We've
Right.
done it for so many years. We have some confidence that
Yeah.
this code is not totally
The code is smarter than we are, at least me. So
Definitely
this
me.
got yellow before. We talked about that. I just failed to map this picture onto how at any given pixel you have this decrease in the short wavelength, but it's quite important. And then the other thing you can see is that the modulation up and down is much less at the shorter wavelength, and it's just growing as the point spread function goes from polychromatic aberrated here to fairly sharp, and the modulation gets bigger. And if we could rotate this around, maybe I can do.
Yeah, do that. That's good.
I will see that the modulation up at the longer wavelengths becomes less than it is in the middle, although not quite as bad or quite as demodulated as it is at the shorter wavelengths. So that's quite
it's hard to see that the first time. So spend a little more time there just so you see,
right.
you know,
So
how
here we go.
they overlap one another so much.
Yeah.
At the
Here we are right at the edge looking at the short wavelength modulations. And then if I spin it around,
yeah.
So this is position I've roughly got here. I spin it around. And of course, you could go in and make this plot separately for each wavelength with a little more work, and we may even have a function that does it. But here you see the modulation at the longer wavelength, and then looking past it in the middle, you see that the modulation is much larger. The contrast is much higher, corresponding to this small point spread function in either of these larger ones. So
David, can you relate this to the point spread function? So I would have thought like at that short, at 500 I see. At 400, you have a very blurry image. At 550,
400, it's very, exactly. So at 400, it's really blurry.
Okay.
This particular scale, which I think corresponds actually, it's about 10 cycles per degree in the checks here. I think I've got 20 checks, 10 cycles, 1 degree. It's really blurred out, those checks, because of this enormous point spread function, which I pointed out is almost a quarter of a degree here. Then you get to here, it's about half the size as here, and you spin around and you can see that on the retina at 700 nanometers, you still got some modulation as you slice through these checks. And then in the middle, which is harder to see because it is in the middle, but you can see that if we look from this side, or here I'm now showing the wavelength, basically the contrast is the modulation that you see here relative to the mean, and it's much bigger at this 550 than it is at here. You got to judge the height because the radiance is changing because of the lens, but you get the sense that you see the wavelength dependent blur.
Yeah. Great.
Great. So we'll look in a second what happens
David?
if we were to turn that off. Yes.
I would like to, maybe you can say something about the cos fourth option.
I don't want to say anything about cosine to the fourth.
I think that's why David made the field so small, 1 degree.
Okay.
So so it won't, but it's important for stuff.
I guess we should. So there is, when you carefully calculate a radiance with respect to larger and larger fields, there's a cosine to the fourth fall off with visual angle, which again is in this code. And I wasn't going to say anything about it, but it's there.
A lot of stuff you're not saying things about, which is, it's incredible how much you put in here and save people so much time about having to worry about this. They can go back, and if they have a bigger field of view, then they can look at that effect, right? Yeah.
Okay. So now what I'm going to do is just compute. And again, we did this in the first tutorial. Once we have the cone mosaic and the optical image, we just compute the cone excitations. I'm not adding the Poisson distributed noise to this here, but we're just computing it. And then this particular command is a visualization of the mosaic excitations, which lets us see. Now we're in the fovea, and there are things here that are, again, worth looking at. I'm not going to break it out by cone type, but we can see that, first of all, this 10 cycles per degree is resolved by foveal cones. And you can also see that, again, this is a detail that's important. Cone density and the size of the cones right in the fovea is higher than it is even out at plus or minus half a degree. And in addition, because the cone apertures are smaller, they're absorbing less light per cone, so it's a little dimmer, and there's some macular pigment in there that's going probably across most of this whole thing. These were things we talked about in that earlier tutorial. And here I just want to say, for these conditions, 10 cycles per degree, we can resolve, you can see from the mosaic that this checkerboard is resolved. And what I'd like to do now is—
wait, before you go away from
before I go, yep.
Yeah. Everybody's going to be looking at this and wondering, what the heck are those big black spots?
What are those big black spots? So they're sparse maybe about 5 to 7% of the cones, and they are S cones, and S cones just have a lower excitation rate than the L and M cones, primarily because of this effect that the irradiance down in the short wavelengths is way down from the irradiance at the longer wavelengths, and that decrease makes the just the whole level of the S cone excitations smaller, and we're plotting it here accurately in that regard, in the sense that we're plotting the S cone excitations as smaller, and you don't actually see easily at this scale the modulations which are really there. The other thing you'll note is there are none of those little black spots in about the central 20 degrees. And this is the so-called tritanopic 20 minutes. I'm sorry, minus 10 to 10, 20 minutes, not 20 degrees. Thank you. Tritanopic area, which is typical in the human central fovea, where there are no S cones. The LM cones are at their densest there, and we're modeling that in our default cone mosaic, although again, you can put them back by appropriate control, the C mosaic, which in some future tutorial we'll talk about how to manipulate.
And for which we thank W.D. Wright and Wilmer for having told us that there are no blue cones in the middle over there. No S cones.
No S cones, but yes, at the time, the working group on the blue mechanism helped figure that out. Yeah. Okay, so that's this whole tutorial. I've run the whole thing and shown you the pieces that we can now manipulate. And I think one thing that would be kind of fun to do first is let's move out from the central fovea to 5 degrees visual, 5 degrees of visual angle along the horizontal meridian. I'm just going to run the thing again. It's going to create a mosaic appropriate for that eccentricity, and it's going to use optics appropriate for that eccentricity. And if I, and by the way, yeah, so I haven't changed anything about the stimulus. You probably, if you remember what we're looking at, you can already see this is a little blurrier because the optics get blurry. This is, looking at the point spread function. If you remember, the best focus in the fovea was maybe half this size, so it's blurrier out there. Chromatic aberration sort of dominates at these locations. And these are based on actual data measured at 5 degrees by Follins and Artal that we've clocked in. The other thing you can see, the cones are less dense at 5 degrees. And what I wanted to illustrate here is that you can't really, it's getting quite hard to resolve that checkerboard out there. We know that vision gets worse in the periphery, and that effect is being driven by these peripheral factors to a large extent. Certainly the checks are less salient when we've gone up to 5 degrees, which is a combination of the optical blur being worse, so less contrast, and the cone density and the sampling being less, probably a little more the blur, but so let's go and make the checks bigger and just make sure we haven't broken anything. That would probably be reassuring. And again, this is something when I work with this software, I often, it's easy to make mistakes, but okay, good. Made the checks nice and big. And now we can see a couple things that are kind of fun. One is we can see that the mosaic now easily resolves these checks, which are basically at 2 cycles, or really 1 cycle per degree. So I made them 10 times bigger. The other thing you can see really clearly, because the edges are sharp, is the chromatic fringing that's produced by the chromatic aberration. So you see that there's the red and the blue are not of the same width. They've spread out relative to each other. And so you see a little more blue. You can't quite see it over the black. It's just kind of dark, but the short wavelength stimulus at these edges has been blurred out more. So that's a consequence that you can see in the retinal image.
Hey, why don't you do one of those plots, like go to analyze, and let's see if we can see it in that. Yeah. Okay, so let's see. Here's position, here's the edge, and what we expect is that that thing is going to take more time going from bottom to top as we look down the barrel of the edge, which I ought to be able to do Yep.
this way. And there you can see it, right? You can kind of see like the little sand dune is spilled out more. That's the, in some sense, it's the line spread function. That's spilled out more here, or the edge function, than it has in the middle, and then not probably lined up quite right. But you can, if we spun around, we could see the same effect going from long to short. But actually, this is a better view because now we can see that when it's coming down and it tucks under, and then the short wavelength is a little blurrier. So you can really see that happening here.
And our buddies, Marty Banks and Steve Choliak, very clever.
Choliak, yeah.
You know, really demonstrated that this phenomenon in the retina has an impact on how it is we accommodate. It's because this kind of effect is a signal in the eye, and the brain uses it, and that'll be a topic for how that happens in another.
Right. I should say that I think before those guys did the experiments, which showed it, Bill Geisler and Johannes Berge did some calculations showing that there was information there. And again, in these kinds of simulations, if you want to check out how well does it work, what happens, we're set up the same. I now want to show what happens, and again, I'm going to now turn off the human eye's chromatic aberration in the way I described to you. And now I do this with my class. What do you think is going to happen? Well, we better not see very much change and chromatic fringing anymore if we've turned it off. And there's still a little bit of wavelength dependent change due to diffraction effects. But now, okay, great. First of all, this is not as severe. And we can see that in the point spread functions. And actually, I think the diffraction effect may even, this is actually these axis scales. I should have scaled them the same. I'm a bad person. This one is actually a little more compact at the short wavelength because the effective diffraction on wavelength goes the other way. And we can turn that off, but it's a real physical effect that probably should be modeled. But if we went back to this graph here and again did our little game, I think now we would, and again, you have to try this at home if you don't remember and quite how these plots looked, but that really pronounced, you can certainly see it's changed. And if anything, it's sharpest here and at its most gradual here, but these effects are really small in the point spread function compared to the effects of chromatic aberration. And then we might wonder something like, well, suppose that I got really good optics for the human eye. I turned off the chromatic aberration. Now, could I resolve that checkerboard back? I'm going to do two things at once here because we're sort of getting a little long. And so I'm going to put the check size back at the 10 cycles per degree. I'm going to stay out at 5 degrees. I'm going to now model a diffraction limited human eye. So the point spread function is going to be better without chromatic aberration. And I'm not going to add any defocus. And I'm going to run that.
So this is really just kind of an ideal optical system. It's
this is about as ideal. Yeah.
Hmm.
And now you can see, first of all, this is now really quite a bit sharper. And you can see that, well, there's a pattern there, but the sampling is really making it hard to see that it's a checkerboard instead of a hexagon. We're really just at the edge of, roughly speaking, the Nyquist limit for this pattern here at the 10 cycles per degree out there at 5 degrees. But we can, again, with these manipulations, separate in terms of what are the cone mosaic responses, the effect of the optics, which I've been mucking with, from the effect of the cone density. And I could, I don't know, I could go in either direction here. I could go further out and really show it mucked up. Maybe we'll do that.
Yeah, so your point here is that some of the information is lost because of the optics.
Yeah.
And some of the information is lost because your cones are getting so big and the spacing is large.
both of those are my points. And in addition, I just wanted to make the point that, with software like ISETBio, because we have these pieces and we can turn them on and off, if you're interested in understanding things about the relative contribution of the different pieces, you can turn them on and off and calculate whatever you wanted to calculate with the mosaic responses and explore these things, which is something we do quite a bit of.
Right.
Okay.
Your slides also had kind of a nice demonstration that the visual system is expecting a blurred image in the short wavelengths, and so that's why the
yes,
slow sampling.
if we went and looked at the S-cone submosaic, and we actually asked about what the effect of chromatic aberration would be, it does prevent aliasing by the S-cone, spatial aliasing, because it removes the contrast before you hit the aliasing frequency. All right, so I'm going to do maybe one more. Anyway, this script is here. We've gone through it, and something I think if you're interested in this, it's there for you to play along with at home and to explore. But it might be fun to do one other thing, which is to go back to our original situation. I think you can try adding some defocus. I'm not going to do that here live, but if you set this to half a diopter or a diopter or a quarter of a diopter, you can see what that will do to the optics, either diffraction limited or human optics. But let's just look at one more thing. I'm going to run this again, and then I'm going to change the subject from the Poland's data set, from the one whose point spread function was sort of the best to the worst, and we'll just kind of look at what that does, which I'm honestly not sure I've done. So I'll be as interested as everybody else. So this is where we first one that we ran, and then I'm going to go down. Where is that subject specified? Oh yeah, rank order. Well, again, I think I got this code right, so that if I go to, let me just go to 10. I think is the number that there are, and scene is going to be the same. Oh, it's definitely, whoa, okay, we really blurred it quite differently. And let's take a look here. This subject has got rather astigmatic optics and rather bad optics. So you can see, first of all, even at 550, they have a point spread function which is asymmetric, so that the blur in one dimension is going to be more than the blur in the other dimension. You also see that, and this is one of these things that these wavefront aberration measurements just, I find it quite remarkable, how aberrated the human eye can be and is. It's not a terribly regular thing, and you get these aberrations all over the place. You get chromatic aberration affecting things. Sometimes chromatic aberration, adding a little defocus to a highly aberrated eye, doesn't necessarily make the point spread function worse. This is something Steve Burns pointed out in a paper quite some time ago. Often it does, and at the short wavelength, this subject is bad. And now, although we could resolve that checkerboard at 10 cycles per degree for the best subject, it's getting pretty marginal, and actually the checkerboard has been blurred in a way that makes it look more like stripes. So we see this large individual variation in subjects. I can't resist, and then I think I'll stop unless people have other things they want to add. I'm going to go halfway in between to see what happens.
And one of the things many of us have wondered about over the years is, suppose you grow up with one or another kind of optics, and it's there in your eye. Can the ganglion cells and cortical cells do anything to compensate for these kind of relatively fixed blurs at the front end?
And I think, I think again, it might be Pablo Ortal and Dave Williams, and possibly I think it's Jun Young. What is his name? I'm sorry, Jun Young. Yoon at Rochester have actually used adaptive optics to either correct for people's optics or to dial in to person A the aberrations that were measured for person B. And one of the things you discover is that everybody's vision seems to be better for their own aberrations than somebody else's. And don't know why that makes me think of the movie Spinal Tap, but never mind. And better for their own aberrations than somebody else's. And
Lost me.
yeah, never mind. It's a little off color. It's suggesting that there is a kind of eye-specific adaptation that goes on. How much it, now can't put back information that's not there, but it can do some degree of image sharpening or contrast enhancement or whatever else should happen. Anyway, for this intermediate subject, you see that there are the checkerboard has kind of come back. And also, again, you can see the chromatic aberration on this particular one has caused a substantial offset. So
yeah, the
I think that's,
astigmatism.
yeah, and yeah, a little astigmatism here. So I am going to stop there. And
Just one question for the subject 10, when you show a checkerboard, does it appear to be stripes in oblique stripes to that person, or getting to your point?
I don't have access to subject 10. I wish I knew.
Right.
Yeah. But it would be it would be interesting to know. In part, it would address some of what
yeah, what you were just talking
Brian
about.
just asked, that it might be that information has been lost, but that appearance has been, at least for suprathreshold things, has been preserved. Certainly, we tend not to experience large chromatic fringes under most circumstances, suggesting that the visual system has adapted to the chromatic aberration of its individual eye, at least to some extent.
Mm-hmm. Yeah, be an interesting psychophysical experiment.
Well, great. And, David, the other thing that really comes through for me when you present this is the, here we're looking at the optical image and we can resolve it and so forth. And how much of the thinking about human now further depends on where this image is placed on the retina.
Yeah.
On the fovea, it's, we're looking at it, we think, oh, it's there. But you know what? When you put it on the periphery, you can't pull that apart because of the size of the, that's good what you're doing. You can't pull it apart because of how drastically the sizes of the photoreceptors change just over the central couple of degrees. Yeah. And therefore, how much more quickly the code runs, because you have so many fewer cone samples 5 degrees out.
By the way, I think if you comment out these commands up here and run it again, it won't close the figures, which might be useful. But in any case, perhaps we should stop the recording and call it a wrap.
Yeah,
Okay, thanks, David.
Thank you.